Welcome, fellow learners. If you are facing that monumental task of mastering human physiology, you know you need the highest yield information, just distilled perfectly.
That's the goal.
Today, we are taking a deep dive into the absolute core concepts of human function. We're pulling crucial clinical nuggets directly from the essential Guyton and Hall Physiology Review, 3rd edition.
And our mission today is really to move beyond just reciting facts. We're here to distill these complex, interrelated concepts across multiple body systems, from a single cell all the way up to systemic control, into concise, applicable knowledge.
So we're getting the scientific clarity, but also the practical so what context.
Exactly. That's what you need for true mastery.
Okay, let's unpack this across the major systems then.
Yeah,
I think we have to start where all physiology begins, right? The inner workings of the cell.
absolutely. And inside the cell, let's begin with energy. Everyone knows mitochondria are the powerhouses.
Right, that's the classic.
It is, but the critical detail is how much energy they're actually making. We're talking about approximately 90% of the total ATP from glucose metabolism. It all comes from oxidative phosphorylation in these organelles.
So if the mitochondria fail, the cell is just left with these really inefficient backup plants.
It's basically running on fumes, and while the cell is making energy, it's also, manufacturing and detoxifying. Which brings us to the endoplasmic reticulum, the ER.
Another powerhouse, but for different things.
A powerhouse of synthesis, for sure. It's the primary spot for making proteins and lipids. But here's the function that I think often gets overlooked. It provides the enzymes for breaking down glycogen and, crucially, for detoxifying things like drugs.
Okay, so manufacturing, detox, but then you also need waste management. And that's the job of the lysosomes. We found a really specific clinical connection here that shows just how fragile this system is.
Yeah, lysosomes are essentially the cell's digestive system full of acid hydrolases. And a perfect clinical example is a deficiency in acid lipase A. When that one enzyme is missing, you get this pathological buildup of cholesterol esters and triglycerides.
And where does it build up?
In key organs, like the liver and spleen. It's a dramatic reminder that if you can't break down the trash, you can cause systemic organ failure.
So from the inside, let's move to the outside of the cell, to its identity, the glycocalyx. This isn't just some sticky coat, right?
No, at all. It's a very specific identification layer. The glycocalyx is this negatively charged layer of carbohydrates attached to proteins on the outer surface. Think of it as the cell's fingerprint.
Which is fundamental for what? Cell-to-cell talk.
For all of it, cell interactions, binding to ligands, and really dictating the normal immune response. Your immune system uses it to tell friend from foe.
That structure dictates function, which includes how cells grow or don't grow. Let's quickly touch on the oncology basics you pulled out, the balance of growth control.
It's a constant tug of war. On one side, you have proto-oncogenes. These are normal genes that code for proteins that, promote controlled growth.
And on the other side?
You have the anti-oncogenes, what we usually call tumor suppressor genes. Cancer often happens when these protective anti-oncogenes get inactivated. That just lets the oncogenes run wild.
Okay, let's wrap up the cell segment with electricity and transport. Because without transport, none of this works. We usually start with that resting membrane potential. What is it? Around minus 90 millivolts in a typical neuron.
That's right. And that negative charge is fundamentally established by the Nernst potential. This depends on two big factors. The membrane is super permeable to potassium, or K+, and there's a really high concentration of potassium inside the cell.
So if you mess with the potassium outside the cell, you immediately change that resting potential.
Immediately. And driving all of this is the engine, the sodium-potassium ATP pump.
The classic example of primary active transport.
It's primary because it directly burns ATP for energy. It's an expensive pump to run, but it's absolutely vital. It pushes 3 sodium ions out for every 2 potassium ions it brings in.
So that sodium gradient it creates isn't just about voltage, it's stored energy.
That's the perfect way to put it. That massive sodium gradient is the energy source that then drives what we call secondary active transport.
Which powers things like
What? Crucial things, like the sodium-calcium countertransport that keeps intracellular calcium low and, critically, the co-transport of glucose into your intestinal and renal cells. If the cell can't make ATP for that main pump, all the secondary transport just grinds to a halt.
That reliance on energy and precise control is a perfect transition into our next section. Regulatory systems. Moving from one cell to the whole body, stability is everything. And that means negative feedback.
Negative feedback is all about the body correcting an error, like your baroreceptors clamping down when your blood pressure drops. The sources even give us a formal way to measure this, the feedback gain. You calculate it as the correction divided by the remaining error.
Okay, so if your pressure drops by 20 points and the system corrects 10 of those points, you still have an error of 10. So 10 divided by 10 is 1. But it's negative feedback, so the gain is -1.
Exactly. And a bigger number, like a gain of -5 or -10, means the system is incredibly effective at restoring balance.
Now, contrast that with positive feedback.
Positive feedback, usually a disaster. It promotes instability, a vicious cycle. A catastrophic example is severe heart failure. The heart gets weaker, so its output drops, which means less blood flows to the heart muscle itself, making it even weaker.
But positive feedback can't be all bad, right? There has to be an exception.
There is a vital one, the nerve action potential. It relies entirely on positive feedback. That initial change in voltage causes some sodium channels to open,
And the sodium rushing in makes it even more positive.
which opens more sodium channels. It leads to this explosive all-or-nothing signal. It's a moment of necessary, controlled instability.
Speaking of controlled,
Yeah.
let's talk about muscle. We mentioned calcium is the trigger for both skeletal and smooth muscle, but they use different receivers for that signal.
A critical distinction. In skeletal muscle, the calcium signal is received by troponin C. That's the protein that binds up to 4 calcium ions, and that binding directly kicks off the whole mechanical contraction process.
But smooth muscle uses a different blueprint.
It does. Smooth muscle contraction is regulated by calcium binding to calmodulin. It also binds 4 calcium ions, but then this complex has to go and activate another enzyme, myosin light chain kinase, which then causes contraction. It's a slightly more roundabout process.
And this is where the clinical picture gets really scary. Malignant hyperthermia. It just perfectly illustrates what happens when calcium control breaks.
Malignant hyperthermia, or MH, is a genetic disease. In these patients, certain anesthetic drugs can trigger defective ryanodine receptors. These are the channels that release calcium from storage in the muscle.
So they just get stuck open.
Essentially, yes. They dump an enormous amount of calcium into the muscle, causing continuous, uncontrolled contraction. The result is a skyrocketing body temperature, the hyperthermia, and just profound metabolic crisis. A tiny genetic defect turns routine surgery into a deadly emergency.
Okay, let's shift focus from muscle to the entire circulatory system. This is where flow regulation is king, and you have to start with the power of vessel radius.
Yes, from Poiseuille's law, it's almost hard to overstate this.
It's the single greatest determinant of flow, because flow is proportional to the radius to the 4th power. So if you constrict a vessel to just half its radius, you don't just have the flow.
Not even close. You reduce it 16-fold. It's an incredible amount of control. And it's why those tiny little arterioles are the body's main tool for regulating blood pressure.
And what about the speed of the blood itself, the velocity?
Well, velocity is naturally highest in the aorta, right, where it's ejected from the heart. But then as that blood spreads out into the huge combined area of all the capillaries, the velocity just plummets.
And that's on purpose.
Completely intentional. That slow speed is what maximizes the time for nutrients and waste to move back and forth across the capillary walls.
So timing is everything. And that's especially true in the cardiac conduction system. We know the signal moves fastest through the Purkinje fibers, but where does the system deliberately hit the brakes?
The slowest conduction rate is found in the AV bundle fibers, right there in the AV node. And this slowness is a critical feature, not a bug. It creates a total delay of, let's see, about 0.09 seconds.
And why is that tiny fraction of a second so important?
It's the pause. It's a mandatory pause that lets the atria finish contracting and empty all their blood into the ventricles before the ventricles get the signal to squeeze. Without that delay, the pump just doesn't work.
And we can see problems with this system on an ECG. The sources give a clear example with axis deviation.
Right, so right axis deviation, where the QRS complex is positive in lead AVF but negative in lead I, that's often a sign of the right side of the heart working too hard, maybe from something like pulmonary valve stenosis.
And a left axis shift would point to a problem on the left side, like from high blood pressure.
Exactly. Left ventricular hypertrophy. Now let's put it all together. An integrated reflex. Standing up. Seems simple, but it triggers a huge cascade.
Right, the second you stand up, gravity pulls blood down into your legs.
Which increases the pressure in those veins and causes a sudden temporary drop in your arterial blood pressure. The body has to react instantly, the baroreceptors fire.
And that triggers the sympathetic nervous system.
A massive sympathetic response. Heart rate goes up, blood vessels all over the body constrict, and you even decrease blood flow to the kidneys to hang on to every drop of fluid.
But what happens when that system fails? Let's talk about the most critical failure mode, cardiogenic shock, that vicious cycle.
Right. In cardiogenic shock, the heart is already weak, so its output is low. This low output means your diastolic blood pressure is also low. And here's the kicker. The heart muscle itself gets its blood supply almost entirely during diastole,
So low diastolic pressure means low blood flow to the heart.
which starves the heart muscle, making it even weaker. And the cycle just gets worse and worse. It's a deadly positive feedback loop.
So breaking that cycle is everything. The therapeutic goal is what?
You have to increase arterial pressure. You have to interrupt that loop. You do it with things like plasma expanders or potent drugs that constrict blood vessels. Critically, anything that decreases central blood volume, like giving a diuretic too early, will make things worse.
Okay, we've seen how the heart manages pressure. Let's move to the system that manages the volume and chemistry behind that pressure, kidneys. And it all starts with glomerular filtration.
Filtration is all about the net filtration pressure, which is this really delicate balance. It's the forces pushing fluid out, mainly the capillary hydrostatic pressure, PG minus, the forces holding fluid in.
Which are the pressure in Bowman space, PB, and the colloid osmotic pressure, pi g.
Right. So let's use the numbers. If you have, say, 50 millimeters of mercury pushing out, but 12 pushing back and 30 pulling back in.
You're left with just 8 millimeters of mercury. That is a tiny margin. It really shows how sensitive filtration is to blood pressure.
It's incredibly sensitive. Now, let's think about hormonal control. Specifically, what happens with too much aldosterone or hyperaldosteronism.
Aldosterone tells the kidney to save sodium, but dump potassium and hydrogen ions.
And that gives you this classic triad of symptoms. Hypokalemia, so low potassium, metabolic alkalosis from losing all that hydrogen. And because you're retaining so much sodium and water, your blood pressure goes up and you get a profound suppression of renin activity.
The sources also made us think about what happens with irreversible kidney damage. Say you lose 75% of your nephrons. What happens to the ones that are left?
The survivors have to pick up the slack. They have to dramatically increase their excretion volume, maybe up to 4 times normal, just to maintain fluid balance. This means plasma creatinine can shoot up to 4 times normal.
And what does that high flow rate do to the nephron itself?
It basically washes out the concentration gradient in the medulla. So the surviving nephrons lose their ability to make really concentrated urine.
Okay, switching gears a bit to metabolism. Let's talk protein waste. Ammonia is seriously toxic stuff.
It is. Ammonia, which gets released when you break down amino acids, has to be dealt with immediately. It's almost entirely converted to urea in the liver. That's the whole point of the urea cycle. If the liver fails, ammonia builds up, gets to the brain, and causes hepatic coma.
All right, a quick lightning round on vitamins, essential for DNA replication.
That would be vitamin B12 and folic acid. They're needed to make thymidine triphosphate, a key building block for DNA.
Needed for blood clotting.
Vitamin K. The liver needs it to synthesize several of the clotting factors.
And for vision.
Vitamin A. Its deficiency is the classic cause of night blindness because it's a core component of your retinal pigments.
And our final nugget comes from endocrinology and this specialized job of the placenta.
This is a really cool point. The placenta does this critical conversion step. It takes androgenic steroids, which are made in the fetal and maternal adrenal glands, and converts them into estrogen.
Why can't it just make estrogen from scratch?
It just can't. It doesn't have the enzymatic machinery. It relies completely on those precursors, and the clinical takeaway is huge. If the fetal adrenal glands are in trouble, estrogen production plummets. It's a major warning sign of fetal distress.
Wow. That was a really comprehensive deep dive. We've hit the high-yield essentials across cells, control systems, circulation, all the way to homeostasis.
Yeah, and we've really tried to establish those connections, how primary transport powers secondary transport, how controlled instability in a nerve is actually a good thing, and how that tiny 0.09 second delay in the AV node is what keeps your heart pumping efficiently.
Thinking about that nuance in muscle contraction, it's amazing. Calcium is the universal trigger, but the entire outcome depends on whether it binds to troponin C or calmodulin. Such a slight architectural difference in the binding protein leads to completely different functions.
That structural control is everything in physiology. And to leave you with a quick challenge, based on what we covered with fluid balance, Imagine a patient gets an IV infusion of a very hypertonic solution, like 3% saline. Before the kidneys have any time to react, what is the immediate net movement of water between the inside of their cells and the outside, and what happens to the volume of that intracellular fluid?
Hmm.
Just think about that immediate osmotic shift.
A great question to chew on. Thanks for learning with us on the deep dive.